Let's speak about the insulin here. So the insulin will bind to the receptor, leading to tyrosine phosphorylation. Then there are two pathways. The first one is called the RAS-MAP kinase pathway. This RAS-MAP kinase pathway will lead to cell growth and DNA synthesis. Then the second pathway, it's called the phosphoinositide 3 kinase pathway. This pathway will lead to glycogen and lipid protein synthesis. The most important one is this, which is GLUT4. GLUT4 will go to the membrane. Leading to glucose entrance. So through this pathway, the insulin will lead to glucose entrance by the GLUT4 transport, which will be transported from the cytoplasm into the membrane, leading to entrance of glucose. It's called what? It's called the insulin dependent glucose uptake. The second one, it's called the insulin secretion by pancreatic beta cells. So the pancreatic beta cells will lead to insulin secretion. How? There is difference here. It's GLUT2 on the surface of pancreatic beta cells, but here it's GLUT4 in adipose tissue and skeletal muscle, GLUT4. But here on pancreatic beta cells, there is GLUT2, which will lead to entrance of glucose through the glycolysis, leading to more ATP. So the ratio between ATP and ADP will increase because of increase of ATP through the glycolysis of the glucose, leading to more ATP will go to this channel to close it. This channel is called ATP sensitive potassium channel. Once there is closure of it, leading to accumulation of potassium inside, leading to depolarization for what? For this channel. Depolarization leading to opening of this channel. What is this channel? It's called the voltage-gated calcium channel, which will lead to entrance of calcium, leading to increase intracellular calcium. Then there is exocytosis of insulin granules, which will lead to insulin will go to the blood. Then this regulation, glucose is the major regulator for insulin release. So the increase in insulin response through oral versus intravenous, which one is faster than the other? Oral is faster than. Why? Because of incretin, release of glucagon-like peptide 1 and glucose-dependent insulinotropic peptide. These incretins will make the response and release of insulin in cases of oral more faster than the glucose. So also there is release of what is this release of these incretins after the meals, and increase in beta cell sensitivity to glucose. The release of the insulin affected by what? By alpha 2 and beta 2. In beta 2 there is increase in release of insulin. In alpha 2 there is decrease in release of insulin. So beta 2 will increase the insulin release. Then we have glucose that will enter into beta cells. Glucose will enter into the beta cells, leading to what? Increase ATP generation through the glucose metabolism, through the glycolysis. Then closes of this potassium channel, which will lead to depolarization to open the calcium channel to enter the calcium. So leading to depolarization of B cell membrane, leading to voltage-gated calcium channel will open, leading to calcium influx then stimulation of insulin exocytosis. There is a more important note here, which is the closure of the potassium. We have a drug called sulfonylurea. So this drug acting by closure of the potassium channel, so it will lead to the same function. Closure of potassium channel leading to depolarization and opening of calcium channel, voltage gated calcium channel to enter the calcium, leading to exocytosis of the insulin. The same function. So here potassium closure and sulfonylurea. Then we have here the structure of the insulin. The insulin is at the beginning is what? Pre-brew insulin. It's synthesized in rough endoplasmic reticulum in pancreatic beta cells. So we said that it's a pre-brew. So cleavage of the pre part leading to what? Leading to formation of brew insulin. The cleavage of the pre part leading to formation of the brew insulin. Brew insulin is stored in secretory granules. Then cleavage of the brew insulin leading to exocytosis of insulin and C-peptide. C-peptide is so important. Because of what? Because it will differentiate between the endogenous insulin and exogenous insulin. What is the difference between them? Endogenous insulin, there is insulin and there is C-peptide. But exogenous insulin, there is lack of C-peptide. So what are the cases that will increase the endogenous part of the insulin secretion? In cases of type 2 diabetes, there is no response to insulin. There is lack of sensitivity to insulin. But insulin will be released in huge amount. So this insulin is formed inside the body. So it will gain C-peptide. So C-peptide will be released with insulin. And cases of insulin secretagogue, like what? Like sulfonylurea. We said here sulfonylurea will go to the potassium channel and close it, leading to increase endogenous insulin exocytosis of what? Of the endogenous one. So the C-peptide will be released with the insulin. And in cases of insulinoma tumor, it's releasing the insulin with C-peptide. But exogenous, it will lack the C-peptide. No C-peptide. So if it's from outside, there is lack of the C-peptide. Then the next one, which is the function binding into insulin receptor. Here there is binding to insulin receptor, and it will go through this pathway. So binding to insulin receptor leading to tyrosine kinase activity, inducing glucose uptake through the carrier mediated transport. What is this carrier? This GLUT4 transportation of this GLUT4 from the cytoplasm into the cell membrane, leading to entrance of the glucose. So here we have insulin dependent tissue. It's called what? Insulin dependent glucose transporters. It's called what? GLUT4. Adipose tissue striated muscle. Exercise can also increase GLUT4 expression. Then there is increase in gene transcription. So the function of insulin, it's called what? It's called the anabolic function. It will build the body. So it will increase the glucose transport in skeletal muscle and also adipose tissue through what? Through GLUT4. You have GLUT4 in striated muscle and also adipose tissue, and increase glycogen synthesis and storage, increase triglyceride synthesis, increase sodium retention through the kidney, increase protein synthesis in the muscle, and this is the most important one, which is increase the uptake of the potassium, increase the uptake of potassium and amino acid, but increase in entrance of the potassium. So insulin contains 2N. So entrance of the glucose and entrance of the potassium, entrance of the glucose and entrance of the potassium. So in cases of diabetic patients, you must correct potassium with given insulin, because insulin will lead to entrance of the potassium, so you must correct the potassium. And you may give glucose with insulin to correct glucose part, and also you must correct the potassium part, the electrolyte, and decrease in glucagon release. Why? Because insulin against glucose, so it will decrease anything leading to release of uptake of glucose, or increase in glucose in the blood. Insulin will lead to increase in uptake of the glucose, and it will oppose anything against it to increase the glucose in the blood, so it will lead to decrease glucagon release, to decrease the glucose in the blood, because insulin against anything that will lead to increase glucose, leading to decrease lipolysis in adipose tissue. Unlike glucose, insulin doesn't cross placenta. This is so important. Why? Because of infant of diabetic mother, so the glucose can cross the placenta, insulin doesn't cross the placenta. So infant of diabetic mother will suffer from hypoglycemia after the birth. Why? Because during pregnancy there is glucose will go through the placenta to the fetus, leading to hyperplasia of what? Of the beta cells in the fetus, beta cells of the fetus. So once there is delivery, there is hypersecretion of insulin, leading to hypoglycemia for the infant. So this is the patient, infant of diabetic mother, suffering from hypoglycemia after the birth. So we have here insulin independent transporters. We have GLUT1, we have GLUT2, 3 and 5, and sodium glucose co-transporter. So in GLUT1 we have RBCs, we have brain and also cornea and placenta, but the most important ones RBCs and brain. GLUT1, GLUT2, this is in beta, the beta islet cells and liver and kidney and GI. The most important one, beta islet cells. We spoke about it. Here there is GLUT2. GLUT2 on beta cells, leading to uptake of glucose, which will lead to glycolysis and increase in ATP. Then we have GLUT3, brain and placenta. We have GLUT5, fructose is spermatocytes. So of course the GI tract, but spermatocytes because of fructose. So GLUT5 is the most important one. Then we have sodium glucose co-transporter, kidney and small intestine. The most important one is kidney, because there are many drugs. There are drugs that will work through this co-transporter, sodium glucose co-transporter. So you must remember sodium glucose co-transporter through the kidney. Then the brain will prefer what? Will prefer glucose, but it can use ketone. It can use ketone bodies during what? During starvation. So in starvation, brain can depend on ketone bodies, but the main one for the brain is glucose. We have here RBCs utilize only glucose. It will not depend on ketones. Why? It's in mitochondria. This is the most important one. This information is so good. We have here lacking of mitochondria in RBCs. This is so important. RBCs doesn't have mitochondria. We have here bricklets, insulin independent glucose uptake through what? Brain, RBCs, intestine, cornea, kidney, liver, islet, beta islet of pancreas, and also placenta, spermatocytes. This is insulin independent glucose uptake. So what is the insulin dependent glucose transporter? In what? In adipose tissue and striated muscle, and it's called what? It's called GLUT4.